So good evening, everyone, and welcome to the 52nd webinar in the Archaeological Sciences Center's webinar series at IIT Gandhinagar. And as always, we are gathering once again on the last Saturday of the month. This is the first month of 2025, and we begin this year with another wonderful talk. This time we have Professor Eleanora Reber, all the way from North Carolina in the United States of America. She's an archaeochemist and has many, many years of experience in archaeochemistry and specifically organic residue analysis of ancient ceramics. Professor Reber is, of course, a wonderful collaborator. She has been working with one of our PhD students, Ahana Ghosh, on her PhD topic, which involves the organic residue analysis of potteries from several different Harappan culture sites. So when Ahana received the Fulbright Fellowship, she spent a few months in US in the residue facility that Professor Reber has set up in her university and benefited immensely from that experience. Professor Reber has been a wonderful mentor to her and taught her everything, right from organic chemistry to the analysis to sampling and all of it. So we wanted Professor Reber to come and kind of do this talk, which not only talks about, we wanted her to kind of touch upon the fundamentals of this topic and kind of introduce us to the whole process, right from sampling to analysis to interpretation. How does it go about? And maybe also talk about some of the results that have come up from Ahana's work with her. So I request now Purva to introduce Professor Reber to our audience, and then we will begin the talk.
Thank you, ma'am. Good afternoon, everyone. Welcome to the 52nd lecture of the ASC webinar series. It is my privilege to introduce Professor Eleanora Reber. She is the director of the Pottery Residue Lab at the University of North Carolina, Wilmington. She has authored the book An Archaeologist's Guide to Organic Residues in Pottery. Her research interests include archaeological pottery residues, the coastal archaeology of North Carolina, agriculture and plant domestication, archaeobotany, mass spectrometry, stable isotopes, American bottom archaeology and Mississippian emergence, and the archaeology of African diaspora, especially in the Wilmington area. Her lab, the UNCW Pottery Residue Lab, is a dedicated facility for GCMS analysis of absorbed and visible pottery residues. For today's webinar, Professor will deliver her talk on organic residues, residue analysis and sampling and interpreting lipid residues in archaeological pottery. Before we delve into the intriguing discussions, I extend my heartfelt gratitude to Professor Reber for taking out her valuable time to share her expertise with us. Ma'am, you may please begin with your presentation. Thank you.
Okay, thank you, Purva and Professor Sharada. I really appreciate that. So what I'd like to do today is just talk a little bit about organic residues and as archaeologists and as people who are interested in, interpreting the past in general. So a lot of people kind of misunderstand organic pottery residues and how they can be used and interpreted in terms of, ancient life, which is what we're interested in. So first, I want to give a brief explanation of what organic residues are. So basically, organic residues are all the things left over from items processed in ancient pottery. So you notice I'm being careful. I'm not saying it's from things cooked in ancient pottery because, of course, many things can be processed in pottery. People can make perfumes, they can make resins, they can make dyes, they can make very, very many things that are not food. That being said, mostly it is food, but not always, an important thing to remember. Since these are remnants of things processed in the pots, kind of the key element in terms of the residues is what will first get out of the contents of the pot and into or onto the fabric of the vessel, and what will last in the archaeological record, and how it will last. All these things mean that residues are not exact templates of what used to be in the pot. Some things don't absorb in the pot at all. some things get washed out again in the archaeological record. And so as a result of this, most of the time, not always, but most of the time, those compounds that survive in the archaeological record are lipids. The reason for this is that lipids are just enough hydrophilic that they'll basically dissolve in water, as in cooking liquid, or as in other liquids in the pot contents. So then they can get out of the pot contents and into or onto the pottery vessel. But they're also hydrophobic enough that they won't wash out in rainwater, which is a key point. So I just gave you here a few kind of pictures of the different kinds of compounds that very often absorb in archaeological residues. So this here is a wax. These are this up here is a alkanol or a long chain alcohol. These are sterols and terpenoids. And we have also a triacylglycerol, which is basically a fat or acylipid, and a fatty acid. As you can see, the triacylglycerol is made up of 3 fatty acids stuck on a glycerol backbone, hence the name. And we will be talking a lot about fatty acids because most fats and oils in cooked or processed items are in this form. And that means very much the most common compound we ever find in a residue are these fatty acids. So that's the compound we find the most of, although it is not interpretively really the most precise, as we'll talk about. So just some terms. Visible residue or residues can come in kind of 2 flavors. The ones you can see on the surface of the artifact, which I call visible residues, and so you can see it right there. It's visible. So those are those things that are deposited on the pot. Sometimes maybe someone might burn the food or burn whatever it is they're making in the pot, and then you see these incrustations or food crusts, as they're sometimes called. So the way these work, it seems like the charcoal forms a matrix on the inside of the pot, and the compounds, the fatty acids, all those other compounds and more will preserve in that charcoal and kind of stay there. Now, what's interesting about visible residues are that firstly, you can see them as an archaeologist, so it's very obvious. And secondly, because the compounds are preserved in the charcoal, it protects a wider range of compounds, not just lipids, but sometimes some other things as well. They also, as archaeologists, can be a very useful source of other kinds of microfossils, as in starch grains, phytoliths, and pollen. So if you've got a visible residue, you can do the residue analysis, but also these other types of analyses. They're also kind of rarer in many cases. More common, but harder to spot, are the absorbed residues. So absorbed residues are those that absorb within the pot fabric, within the unglazed pottery, and you can't see they're there as an archaeologist. We do not know until we test. As a practical matter, it's pretty much mostly lipids that absorb within the pot walls. And these lipids are protected by the little holes that exist in unglazed pottery. So this sort of analysis is really most effective on any kind of unglazed pottery vessel. If you've got a glazed pot, it does not work. You have to have visible residues or no residues. And in general, although we don't entirely, it's kind of embarrassing, understand the mechanism by which they're absorbed residues are kind of an average of all the items ever cooked within a pot. So with a visible residue, someone can burn one meal, and we're seeing maybe the remains of that one kind of bad moment in cooking. But in absorbed residues, we're seeing chemicals and lipids that absorb from most or all of its uses. We're just not sure if we're seeing more of the early uses or the late uses, but it's certainly more than one. So I just want to talk a little bit about how these residues and how these compounds are absorbed and preserved, because it's a key point here. So we have all the items that might be processed in a pot, whether they be food or not food. Everything over time is cooked within the pot and then absorbs within the pot, from the water. The pot breaks and is buried. The pieces of it are buried. Some compounds wash out straight away, the ones that are that easily dissolve in cold water. Depending on it's very dependent on the soil chemistry, the rain chemistry. It's kind of a subtle and complicated system. But in general, after about 6 months or even less, we'll begin to see a range of transformations within some of the compounds that are still absorbed within the matrix of the pot. So some, but not all, of these compounds will undergo oxidation. Air will get in there. Some will undergo hydrolysis. The water and the groundwater will begin to break apart, for example, triacylglycerols, to give us just the fatty acids. And then some microbes, bacteria and so on, maybe even little funguses, will get in there and start to break down those compounds. So that means that when we analyze the contents of our archaeological pot after, many years of burial, we don't see exactly what went into it in the first place. We see everything that survived the first step, and we survive the results of whatever happened to those compounds while they were in the ground. So many of these compounds survive, and interpretation is possible. But what you cannot do in residues is take a modern resource and compare it directly to an archaeological resource, because the chemical transformations are much too great. They don't look much alike at all. I also want to put in a little caution about dietary reconstruction. Obviously, if we're doing pottery residues, we can only analyze the things that went into the pot, right? But people eat a lot of things that are not cooked in pottery. So, for example, anything you cook on a griddle will generally not absorb within a pot wall. Anything that people cook or roast on the embers with no pottery at all, we won't see it. So pottery residue really tells you what people are using their pottery for, which is not the same as what they ate. This is a key point that people often forget in their eagerness to analyze pottery. Another key thing to remember is that since most of the residues are lipids, those foods or resources that have more lipids in them will preserve better in the residue, right? So that means that fattier resources are easier to reconstruct than less fatty resources. So that means, and to work out which things are liable to be better preserved, we have to look at the fat content of the various items. Broadly speaking, not always, but broadly speaking, plant-based resources have fewer fats than animal-based resources, unless you've got a very oily plant resource like nuts. So nuts, for example, are fattier than very many animal-based resources. So that means that, say you've got a mixture of perhaps wheat or rice and maybe dairy. The dairy will come through better than the wheat or the rice because there's much more fat in the dairy food than in the wheat or the rice, which are very low fat. But if you had a mixture of dairy and nuts, then the two would maybe come through about the same because they're both very fatty resources. And of course, since we're only looking at things that were cooked in pottery, variations in how people cook foods from region to region will affect what's preserved. So if people are cooking on the inside of an oven like this, it's unlikely that much residue will preserve on the oven, even though it's an unglazed pottery oven, because there's very little cooking liquid to mediate between the surface of the bread and the wall of the oven. So I hope this is making sense so far. So one of the real stumbling blocks in residue analysis is trying to figure out exactly what was cooked or processed in a vessel. And this is tricky because from a chemical point of view, very many foods are very similar. They all have triacylglycerols. They often have a small little range of waxes. And of course, sterols. So meat often has cholesterol, dairy often has cholesterol, fish products, similarly cholesterol, whereas plant derived foods have different plant sterols, as in cytosterol, camposterol, but they're much less abundant than the triacylglycerols, which are pretty common and are present in most resources. There are some compounds that are unique to different resources. We call them biomarkers, but they don't always survive in the archaeological record, which makes our job tricky. And as I said earlier, foods lower in fats tend not to be absorbed within the vessel as well, and so are not preserved as well. Sadly for us, this includes almost every staple grain, except corn, actually. Corn has a fair amount of lipid in it. But even so, corn has less lipid than many animal products. So, for example, if one's looking at wheat, at rice, at millet, all of these staple grains are low in fats and will tend to be preserved less well in residues. So I said that some compounds were unique to different foodstuffs, and these are very important to us as residue analysts. These are what we call biomarkers. So a biomarker is any compound that's unique to a food. So for example, if we find caffeine in a residue, caffeine isn't a lipid, but sometimes it preserves, that will tell us it's from one of the very few foods around the world that has caffeine in it. So in Asia, that might be tea, for example. In Africa, it's more likely to be coffee. In North America, it's more likely to be yaupon. In Mesoamerica, it's more likely to be chocolate. But there are very few foods around the world that have caffeine. So if we find it and we're sure it's from the archaeological resources and not modern contamination, then we have a pretty good idea of what was in the pot, at least part of the time. As I said, the more common biomarkers are what we call category biomarkers. These are compounds that are present in a limited range of resources, as in cholesterol in animal products, cytosterol in plant products, and then resins. Resins are very highly distinctive. So dihydroabiatic acid is the most common category biomarker for resins from usually conifer trees. There are different category biomarkers and sometimes even resource-specific biomarkers for other resins. Birch, for example, is highly characteristic. But these biomarkers that are unique to a specific resource are quite rare. Category biomarkers are more common and can be very useful in our analysis of what was in the pot. And this picture here is just the mass spec of dihydroabiatic acid, which is that biomarker for pine resin that I mentioned. And here's a different resin biomarker there. We also have compounds that we call indicators. So these are still not completely common, and they're mostly present in particular resources, but not uniquely so. So perhaps the most famous of these are isoprenoid fatty acids. So these are two different isoprenoid fatty acids. Here's pristanic acid, and here's trimethyl tridecanoic acid. But we can just see by looking at these molecular structures that they're very branched, right? They're very regularly branched. They actually derive from the breakdown of chlorophyll, so they can occur in a wide range of resources, but they mostly occur in fish and shellfish resources because the marine diet is essentially based mostly on algae, and algae has a lot of chloroform breakdown in it. So usually, if you find these fatty acids, we view them as an indicator for fish or shellfish, but these compounds do occur in other resources. So they're not a biomarker. If I see it, I can't say, aha, definitely fish. I can say this is one indicator for fish. But these compounds can appear really in any resource deriving from chlorophyll breakdown, including, unfortunately for us, sometimes dairy. Prostanic acid specifically was originally discovered in butterfat, so we know it must be present in dairy. And indeed, in any resource that has any sort of chlorophyll breakdown, it's possible for it to occur, just not usually in the quantities that's present in fish or shellfish. So for resources with indicators like this, and fish and shellfish are probably the best known of these categories, we look for at least 2 or 3 indicators to be a strong supporter of having fish or shellfish be processed in our pot. Just one, it indicates it, but it's certainly not definite. So I had said that triacylglycerols were the most common resource, or the most common lipid, and that triacylglycerols were made up of 3 fatty acids. And so that means that fatty acids are the thing we find the most in residues. So I would say when you're doing residue analysis, you always get fatty acids. And in fact, it pretty much always includes the 2 most common fatty acids, which are the ones with 16 carbons and the one with 18 carbons. No double bonds, no branching, just straight up palmitic and stearic acid, which in residue analysis we usually refer to as C16O and C18O. So these 2 fatty acids occur in like every food residue. Everything you've eaten today has these 2 fatty acids in it. They are very, very common. So that means they're not very helpful when we find them in a residue. They just tell us something was processed in the pot, which may be useful if we're interested in whether the pot was used at all or was used for lipid-containing resources at all. But it doesn't help us identify usually what was in there very carefully or very much. That being said, since biomarkers and indicators are rare, but fatty acids are common, we do use them carefully as sort of a broad indicator of sort of the overall contents of a pot. But it's risky. It's more subtle, and you have to be careful with it. That being said, in an animal-based resource, as in dairy, they nearly always have more saturated fatty acids, and they usually have more C18 stearic acid than C16 fatty acids. Plant-based resources usually, but not always, have more unsaturated fatty acids and more C16 than C18. And then fish or shellfish usually have longer chain fatty acids, between 20 and 24 carbons, and usually with multiple saturations, which is why fish and shellfish go bad so quickly. It's all their polyunsaturated fatty acids becoming hydrolyzed and oxidized and so on. Now, this is where you have to be very, very careful. That's why I say it's a subtle approach. You can't just take, some milk and do this analysis on it and look at it and say, aha, this must be, animal. Because if you took fresh milk from today and analyzed it, it would come out looking like archaeological plant because of all that breakdown, the hydrolysis, the oxidation that I talked to you about. Instead, what you have to do is look at the results of that milk, if it was in a pottery vessel that was buried for a while, and see what effect that had on the compounds. So here, because I've worked mostly with corn in my life, I show you, it's called a gas chromatograph of fresh corn extraction. So what in this sort of graph, what we're seeing is each compound in the residue shows up as sort of a peak here and is labeled. So C16O is that C16 fatty acid I told you about. C18-2 is an unsaturated 18 carbon, and you see there's a lot of it, right? Very much. And if we look at this as well, we can see that there is some C18O, but it's very small. See, it's all the way down here. So it fits the description I gave you, right? There's more C16O than C18O. There's more unsaturated fatty acids. And there are as well a variety of other compounds, including these plant sterols, which are biomarker for plants, camphosterol, stigmasterol, and cytosterol. And here are the triacylglycerols. Since it's a fresh residue, there's still lots of these, fats in their sort of normal state. All right, now here's that same corn residue after it's been buried for 6 months, in this case in a clay loam soil. That looks really different, doesn't it? What happened to all our unsaturated fatty acids? Well, they all got eaten by bacteria, pretty much. And look what happened to our C16O versus C18O. There's less C16O than 18O, which is, disappointing to us. There is still plant sterol right there, but only one. We've lost 2 of our plant sterols. We still have some triacylglycerols, but they're, lower, less of them because they've been breaking down through time. So we see from looking at these 2 graphs that a fresh residue looks really different from an archaeological residue. And that's why we must be so careful about using fatty acids to interpret residues overall. So honestly, if I were to interpret this, not knowing it was corn, I might say this is mixed because the fatty acids look a little bit like a animal product, but it clearly has plant sterol. So I would say definitely plant present, but maybe something else, I don't know. The kicker, of course, is that we also have these compounds. So if I were to chemically break apart these triacylglycerols and these diacylglycerols to remove the fatty acids, our fatty acids here would begin to look more like corn, more like fresh plant. So we would have more C16O and less C18O. And we would probably begin to see these unsaturated fatty acids again as well, because they would be protected on the glycerol backbone. So if I were to do this, chemical breakdown of the acyl lipids, which I do when I'm doing my extractions, then it would again look like a plant, and I would interpret this as completely a plant residue. But again, we must be very cautious when using these fatty acids. And of course, there's no rule that people in the past only use their pottery for one thing, right? They're likely not to. So it may be that a vessel was used for many different plant foods, or a mixture of dairy and animal products and plant, or, maybe a mixture of fish and shellfish. And so it is very possible for a residue to include multiple resources of multiple different types. All right, so that's residue analysis. Let's take a deep breath. If you have any questions, maybe store them up for later, because now I'm going to talk about the next step. So as we look at residues, some of the things we're very interested in are, of course, staple grains, and of course, resources like dairy, milk and cheese, and so on, yogurt. But to get at these, just residue analysis will not be very helpful, because most staple grains are, as I said, not very distinctive and don't usually have much in the way of biomarker. Millet actually does, but it's not very common, and no other staple grain has such a biomarker. But if we pair stable carbon isotope analysis with residues, we can do these things. So we're going to get a little bit technical here. And I sort of apologize, but what we're going to do is talk about how we can use compound-specific isotope analysis to identify resources such as millet or other C4 plants and dairy, and in some cases, marine fish. So fish from the ocean, not fish from lakes and rivers and so on. So in essence, when we do compound-specific isotope analysis, we take our gas chromatograph, as we have just here, but instead of using it to identify all the different compounds in the residue, we use it to get the different stable carbon isotope of each compound. And we can do this because, as I showed you, each of these lipids is made up mostly of carbon, right? So it's all carbon, a small amount of oxygen, and a little bit of hydrogen is the typical atomic makeup of lipids. If it has carbon, we can do carbon stable isotope analysis on it. And that gets us into this very subtle and complex kind of science of stable carbon isotope analysis. So the technology used for this is called gas chromatography, isotope ratio, mass spectrometry, but we all call it GC-IRMS for short. So as I say, we can use this to look at situations where we're seeing foods with different isotopic signatures, which means we're going to have to talk about what gives these isotopic signatures. But let's just look at this graph. This is again of corn, because that's what or actually of an archaeological residue, not from South Asia, but in this case, actually from Louisiana in the United States. And we can see, as we look at the different alkanols, that most of these have these values in the -30 parts per mil. But this one here has quite a different value at -21.9 parts per mil. This compound derived from a resource that's isotopically different from the others. In this case, it derives from corn. Since this compound is very abundant in corn and quite rare in other resources, the isotopic signature of corn will show up much more brightly with this one compound than with the others. And so we can use this compound to identify the presence of corn in a pot that was used to process a variety of different things, not just corn, but many other plants. But the process can also be used for other isotopically unique resources, including for South Asia, millet and dairy, as well as marine fish and shellfish, as I said. So I didn't know how much you all knew about stable carbon isotope analysis. So I'll give you just a real brief primer on how it kind of works very quickly. So carbon has 3 isotopes. C4 plants are, we'll get to it in a minute, don't worry. So carbon has 3 isotopes. One, of course, is radiocarbon, but it has 2 stable isotopes, meaning that they do not break down over time radioactively. One is 13C and one is 12C, the most common. So here's carbon-13. It has 13 neutrons in its nucleus here. And here's carbon-12. It has 12 neutrons in its nucleus here. Carbon-13 is heavier than carbon-12. It reacts exactly like carbon-12 in all other ways. It has the same number of electrons, but because the nucleus has 13 neutrons, it's just a little heavier. And what's interesting is that although it doesn't make much of a difference in kind of wet chemistry, in terms of biochemistry and photosynthesis, it makes a difference. So that means that depending on the sort of photosynthesis a plant has, carbon-13 will be processed differently and will be incorporated into the plant tissues differently. Furthermore, various carbon sources around the world, well, there are really only 2 primary carbon sources around the world, have different amounts of carbon-13 and carbon-12, and that's reflected up the food chain. So terrestrially, on the land, the carbon source is carbon dioxide from the atmosphere. All food, all plants from the land suck their carbon out of the atmosphere from the carbon dioxide, photosynthesize it, and make their own leaves and their grains and, make themselves. Kind of like magic when you think about it. In the ocean, they take the carbon from carbon that's been dissolved in the water over a very long time, some of which is from the air, some of which from limestone and rocks. And as a result, the amount of carbon-13 in the ocean is different from that in the air. And so marine resources and terrestrial resources can be distinguished. Then there's the photosynthesis. So depending on which type of photosynthesis you have, it will incorporate this carbon-13 more or less efficiently, as we say. And for that reason, we can tell what sort of photosynthesis plants have based on their ratio of carbon-13 to carbon-12. And that follows up from the plant to the lipids that absorb in a pot. And it also follows up the food chain through animals. So if a cow, for example, eats some grass, it's getting its carbon from that grass. And then that follows up into the cow itself and then into its milk and so on and so forth. So in terms of terminology, this is how we write this data. Delta carbon 13 is how we say that. And that just means the ratio of carbon 13 to carbon 12. And just because of the standard people picked back in the day, the numbers are always negative. So it's just the way it is. And it's also written in the form parts per mil because we're looking at very, very teeny small differences here. So per mil is out of 1,000, just like percent is out of 100. Okay, so as I say, these two different types of photosynthesis are more or less efficient with regards to carbon 13 versus carbon 12. So here we have wheat. Wheat is what's called a C3 photosynthesizer. This is the very oldest sort of photosynthesis, the very first that evolved way back many millions of years ago. And since this evolution happened when there was more carbon dioxide in the air, back many millions of years ago, the atmosphere was richer with more carbon dioxide. And so apparently the plants could be a little more wasteful with their C13. So with C3 photosynthesis, it's much better at photosynthesizing carbon 12 than carbon 13. And so the ratio changes between the air and the wheat or any other C3 plant itself. So in lipids, which is different from other plant tissues, C3 plants are usually about -28 to -35 parts per mil. Yeah, so that's how that's written, just to like get us ready. So among the, I would say most plants around the world, I'm going to get in trouble for this, are mostly C3. So wheat and rice are both C3. Trees are all C3. Most plants are really C3. That said, there is another sort of photosynthesis that focuses particularly on grasses from very sunny environments. And this is essentially a different form of photosynthesis that evolved from C3 and essentially turbocharges the process. So in C4 photosynthesis, the plant takes carbon dioxide from the atmosphere, sort of sucks it into the leaves, and concentrates it around the photosynthetic cells. This is because over time, the atmosphere has less carbon dioxide than many millions of years ago. And so in some areas, the amount of carbon dioxide in the air has been limiting to the plants. So over time, the C4 photosynthesis evolved, which kind of adds an extra step to the photosynthesis to concentrate the carbon dioxide in the leaves. And this is pretty much exactly how a turbocharger works in a car. So because of this, C4 photosynthesis is more efficient in how it photosynthesizes C13 versus C12. It discriminates less. And that means that C4 photosynthesizers usually are about -9 to -13 parts per mil, more negative than our standard PDB. Millet, for example, is a C4 plant, and actually many of the most rapid growing grasses around the world are C4. So for example, corn is C4, millet C4, sugar similarly C4. So because it's mostly different tropical grasses, it's certainly not as common as C3, but C4 plants tend to grow more rapidly, particularly in sunny environments. And so some very important domesticates are C4. Just to make our lives more complicated, there's a third photosynthetic pathway just for cacti and succulents, but we're not really going to get into that today. Just be aware it exists. So in terms of South Asia, our key C4 resource is probably millet. Oops, sorry. And there are two primary millet types, of course, foxtail and broom corn. Both are C4. Now, when one is using stable carbon isotope analysis, what we can do is use it to watch sort of the advent of plants with different photosynthetic pathways into an environment or a diet. So, for example, in an area that's mostly C3, if they start using millet more, we'll see the carbon stable isotope results change. However, if the environment is mostly C3 and wheat comes in, we won't see a change because wheat is also a C3 plant, if this makes sense. It's really most useful when we see something from a different photosynthetic pathway coming into an environment. So this has been most useful with millet, with corn, and you can actually also do the reverse in China. So in North China, which is primarily C4, when rice comes in, you can see the diet becoming more C3. So it's just sort of a change in the photosynthetic pathway of the primary plants that people ate will be visible. And because carbon follows up through the food chain, this doesn't just work in residues, of course. It's really best known for its use in looking at human diet by looking at bone collagen and bone appetite. But in this application, we're using it to look at the lipids in residues. All right, that was a lot. But one of the things we have to think about as we do stable carbon isotope analysis is that, as I mentioned, there are different possible sources of these different ratios, right? So it turns out, and I'll show you a graph in a minute, that the isotopic ratios of marine fish are very similar to those from C4 plants. We can't just do the isotopes and say, aha, it's a marine fish or it's a C4 plant. We have to base it on the archaeology of the area that the data is from. And in the case of residues, we have to base it on that and also our interpretation of the residue. So that's why it's a bit of a complicated, tricky science. So I've just explained how we can use stable isotope analysis to get at C4 residues such as millet, and also how we can use it to get at marine resources, if that's a likely thing to have. But I said we could also use this data to get at dairy. And now we're going to get even deeper in the weeds, but it has to do with how ruminant digestion works. So just be aware that with residues, and I'll do more with this in a minute, you can use these results to get at ruminant fat, adipose fat, and ruminant dairy. But to get at all these things, we have to interpret the residues because, as I say, the isotopic results can overlap quite a bit. So we need to use everything together to do a good interpretation. We have to interpret the residue and also look at the isotopic data if we think it will be useful at one particular site. Okay, so let's talk a little bit about how ruminants work. As you all probably know, ruminants have a special stomach that allows microbes to partially digest the grass and other plant foods. And this is why ruminants, including cattle, of course, also deer, reindeer, many other resources, but mostly cattle, I think, in your region. That permits them to eat grass most efficiently. Grass is resource-wise very hard to digest. And so it helps to have these microbes in the rumen, hence the name, help digest the grass to get that nutrition into the tissues of the cow. So as part of the microbial action in the rumen here, isotopically, it means that the C18 is more depleted. It has less C13 in it than C12 than the C16O. And this gets pretty deep into the weeds of how the microbes work. For the purposes of residues, I can show you the data, just kind of go with it. So we can measure depletion by subtracting the carbon ratio of C16 from C18. Normal non-ruminant resources are usually about 0, meaning that both the C16 and the C18 fatty acids have about the same amount of depletion or the same stable carbon isotope values. So here's a graph, because this has been a lot of background. And so these green dots down here are our C3 plants. You can see they're on pretty much of a straight line, not exactly, but pretty much. Up here, this triangle, we see the dark triangles are our C4 plants. These red dots here, these are the fatty acids from terrestrial ruminant adipose fats. And we can see they vary a little bit, but they're all sort of down below the line formed by the C3, yes? So this up here is caribou. It's been eating lichen, which is not a C3 plant. It's its own thing. So ignore the caribou. Just keep in mind that this happens. But we can see that that the ruminants are all, on average, more depleted in C18 than our C3 plants and our terrestrial non-ruminant fats. So these blue diamonds, these are the adipose fats of terrestrial non-ruminants. So the ruminants are clustering down here, and we can use this in residues to see if we have ruminant adipose fats. But furthermore, dairy, and the dairy here is our purple triangles, are clustering even more depleted. And this, I'm afraid, gets to how ruminants and other animals produce milk. It has to do with the mammary glands. And as a result of the way the mammary glands work to produce milk, the C18 is even more depleted in the milk of ruminant animals than their adipose tissue. But of course, when humans are involved, we tend to start, feeding different foods to the cows. And the cows eat whatever grass is on hand. And that grass could be either C3 or it could be C4. So these purple triangles up here represent dairy from animals that are eating primarily C4 resources. We can see that they're still depleted, but they're overall have much more C3 in it, or C13 in it, because they're eating these, C4 plants, as in millet or corn or something of this nature. So when we look for dairy in a residue, we're essentially looking for fatty acids that have this kind of depletion pattern, with the C18 more depleted than the C16. And the way this can more easily be measured is, I said, by subtracting the C16 from the C18. In normal resources, we get, and this number is called delta C, I'm afraid, so much Greek. So these resources here, the delta C is about 0. Our ruminant fats, they're, a little bit below 0 to about -3. So -1 to -3. And then in milk, we're seeing below -3.1, with the standard marker for a residue or a fatty acid pattern being largely dairy-based, being anything below a delta C of -3.1. So we can graph this, in this way. And the reason we measure this depletion in this way is simply because of these issues that we don't know what the cow was eating. If it was eating a C4 resource, as in millet, then the raw numbers of the stable carbon isotope value of the fatty acids will be, much higher. But the ratio will be the same if it's dairy. And so here's just some plots of, so here we have up here pig fat. We have ruminant adipose in that -1 to -3.1. And there we have the ruminant dairy down here. We're seeing the same thing here, same thing here, and so on and so forth. So it's a lot of complicated background and a lot of understanding the photosynthesis. But at the end of the day, we can use this technology to distinguish if there's dairy in a pottery residue, or if there is perhaps C4 plant or marine fish in a pottery residue. Okay, hope everyone's still with me. So I'd like now to talk specifically about some data from the site of Karampura that I've worked on with Ahana Ghosh. So we see here, very similarly to our basic graph, it's a little bit differently plotted, but we can see the very bright red dots here are the different residues from Karampura. And here we have the compound-specific data, the C16 and the C18. And we can see some very interesting patterns. Yes, so here's our ruminant adipose. I didn't bother to plot dairy on here because it's clear that most of our C3 resources here are not, in fact, ruminant dairy or ruminant adipose. They're falling right into our C3 plant, not dairy category. So nearly all the residues from Karampura showed up as C3 and no dairy, except for this one. And this is Karampura 186. As we look at it, we can see that it is definitely C4, and it's also like down there, like that's dairy. And so we'll look at the residue a bit. So here's the gas chromatograph of Karampura 186. Here's our internal standard. We put that in to, see how much we had. And as we look at this, our fatty acids, let's see what we see. We have the C16 slightly more abundant than the C18. We have 2 sterile biomarkers, which is nice for us. One from animal products and one from plant products. And we also have that very small little peak with this arrow down here. That's one of those isoprenoid fatty acids that I spoke of, which could be an indicator for fish, but it also occurs in things like, for example, dairy. So if I was interpreting this residue without the benefit of the compound-specific data, I would know it had both plant products and animal products. I would guess maybe there was possibly a bit more plant in it, but I wouldn't be sure enough to make the call. I wouldn't write it in my paper. There's a lot of this small, shorter fatty acid chain, C14. I would wonder if there was fish in there, but there's not. Only one indicator is insufficient. So my only interpretation for this residue without the stable carbon isotope would be a mixture of plant and animal-based resources. But in the presence of the compound-specific data, we can go a lot further. So our delta C, yeah?
ठीक है
Oops.
लेके आ जाए
Sorry?
No, I think someone got unmuted.
Oh, okay.
I've muted that person, so you can go on.
Okay. So we can see our delta carbon 3 is typical of dairy. So we know already that the animal product in that residue is almost certainly milk or, milk product. There's also some plant in there because we had that biomarker. So it's a mixture of dairy and plant resource. There's a strong millet or a strong C4 contribution, which I understand at this site is probably millet of some type. A possible small amount of fish, but I don't think so. I think that isoprenoid fatty acid derived from the dairy in this case. So what we have here is a mixture of mostly milk and some plant contributions. The C4 either came from millet directly processed in the vessel, or the cow that produced the milk may have eaten a lot of millet or C4 plant, leading to that data. Or both, could be both. So by using all these techniques together, we're able to learn a lot more about what was in the pot that we've designated KRN 186. I know we're running a little late on time. Have one more residue I wanted to look at. One of the less abnormal residues from Karampura. So here's Karampura 75. We can see it has a lot more unsaturated fatty acid. It has a biomarker for plant only. It has one isoprenoid fatty acid, pristanic acid, much less C14O. And as we look at the isotopic values, we can get even more data. So this resource does not include dairy. It's probably mostly plant. All C3 contribution, certainly. Possible fish based on the isoprenoid. And since we know there's not any or much dairy in there, then it may be the pristanic acid in this residue came from fish. But again, one indicator is insufficient. So the best we can do is primarily plant from a C3 plants, no ruminant, no dairy. All right, thank you for your patience and bearing with me through all this information today. I know it's been very dense. But what I want you to take away from this is that how you can use residues as an archaeologist and how you can interpret them as a person interested in the past. You can use residues to look at overall resources processed in the pots in general. You cannot use it to distinguish a recipe or anything of that nature. If you've got a primarily C3 environment, you can't, for example, distinguish between rice and wheat because they're both C3. We can distinguish ruminant dairy reliably and marine fish if that's a possibility, but we need to support that through, of course, the archaeological record. Same with the ruminants. In general, residues are really only as good as the knowledge of the local environment and the knowledge of the local archaeological site. And it is complicated, but at the end of the day, residues are really the only way you can get directly at the way that people use pottery in the past, which is a point of real interest to us as archaeologists and people interested, in the way people in the past lived. I'd like to thank everyone here for all their help. I hope I didn't leave anyone out, but I probably did. And to all the people at UNCW who have helped me with this analysis through the years. So yeah, that's what I have. So I guess I'm going to stop sharing and we'll go to the questions now.
Thank you, Eleanora. That was a very, very easy to understand and with all the diagrams and illustrations and step-by-step explanations. I think most of us could follow. I think it's, as you said, it's quite a lot of information to take and would require probably years of practice and understanding to be able to actually use this in our work and make sense of it. But thank you so much for this and also the examples that you provided. So we'll just give there are some questions, but we'll just give a few more minutes to the participants to type their questions. And meanwhile, I have two of my own. So the first question is it possible for a plant to go from being a C3 to a C4 or a C4 to a C3 during its lifetime when the animal's ingesting it or eating it?
Yeah, that's an excellent question. It only happens in one plant, purslane. So I had said that, you had C3, you had C4, and then there is this CAM, crassulane acid metabolism, photosynthesis, which is present in succulents and cactuses. And usually when a plant is one, it just is that photosynthesis and it cannot be changed. But the plant purslane, Portulaca oleracea, it changes back and forth between CAM and C4, depending exactly on the environment, how much rain is present. If it's dry, the plant goes to CAM, and if it's wet, it goes to C4 to be maximally efficient, I guess. So since it happens in one plant, we of course can't be sure it doesn't happen in other less well-studied plants. It doesn't happen in any of the major staple grains, as in millet or wheat or corn. Those are all set. But I would personally suspect that some of the stranger succulents, the purslane type plants, they might. And that's going to add complexity again to our studies. And again, purslane is always a spoiler, as we say. So if you have the potential for purslane at a site, there's some sort of interestingly subtle things we can do to look for it. But if it's there, it could mimic, another C4 plant, depending on the rainfall patterns and so on.
So I just thought of a follow-up question besides the second one that I have, that in this process of human intervention with plants, when we started domesticating them and creating so many variants within a species, right? So have we manipulated this, so as you said, C3 and C4 or the photosynthetic pathway can also depend on several factors like sunshine, nutrients, and the size of the leaves and things like that, right? So through this process of domestication of plants, have we kind of created these artificial environments where plants had to change their natural photosynthetic pathway? And that's why my first question came from it. Is it possible for plants to cross over?
No, not really. Usually what happens is when humans choose a domesticate, we kind of pick it and we change the simple things, the shatteringness, the grain size, the seed coating, and so on. But photosynthesis is usually deeply ingrained within the cellular structure of the plant. So domestication doesn't change it. The ability to shift back and forth for purslane is really unusual. And it's pretty notorious for that reason. But it was inherent in the wild purslane. So if someone were to domesticate purslane, which for all I know they have already, I don't know, it would retain that ability. And they could probably manipulate it based on watering it, but they couldn't add that ability in. So if we domesticate a C4 plant, it stays C4. And if we domesticate a C3 plant, it stays C3.
Good to know that. So at least, I mean, if that was an additional factor to consider while doing this analysis, it would become really complicated. So my second question is, I understand that most of the interpretations are at a broader level, like we say plant or animal or adipose, dairy, and these broader terms. But theoretically, is it possible to have finer resolution to be able to pinpoint and say this particular plant oil or this particular animal? Because we get these questions all the time. Can you tell us whether it was cattle or sheep or goat? Or was it groundnut oil, or was it sesame oil? Or so, is it possible to go, much deeper?
Not usually with lipid residues, unless you have a resource that's got a unique biomarker. Don't know about sesame oil. Might have a unique compound, but most of the time, no.
Okay.
And you can't easily distinguish ruminants. So people have tried, for example, to distinguish sheep from cattle based on the fatty acid, abundances. But the fatty acids break down so much in the archaeological record that this has not really worked very well. Mm-hmm. I think the next pathway forward may be in DNA analysis. I know people have been sort of struggling with identifying chocolate in residues from DNA. You can already do it through biomarkers, but that may be the way forward, but it hasn't been developed yet. And of course, there are contamination issues there as well.
So I see some questions here in the chat box. So we'll take the most pertinent ones. Okay. So the first question is from Anand Kumar, who asks, what is the proper way to collect material from excavation site, let's say pottery, in order for ORA?
Yeah, that's an excellent question. I didn't really get into contamination issues, just because we had so much other things to cover. But yes, contamination is a major problem in residues. Of course, if you're wearing a lot of sunscreen when you dig up the residues, I'll be seeing it. If you dig up the pot, you're covered in the United States, people, of course, are often digging wearing bug spray and sunscreen, and I'm seeing it in the residues every time. And it's not great, really. Particularly a very creamy sunscreen is actively bad for residues. Plasticizers from plastic will just get in everything. It's terrible. So if you're sure you have pottery that you'd like to collect for residue analysis, if you can, try not to touch it all over with bug spray hands or sunscreen hands, and put it directly into maybe tin foil. And if you haven't got tin foil on hand, paper bag is better than plastic, just because that plastic gets into everything.
The next question is from Anirudh Ramanujam. He asks, in terms of glazed pottery, is there any chance for survival of such residues in compared to unglazed pottery?
Sadly, no. And that's kind of a problem, right? Because we've tried on glass, for example. If we had a big trash dump, like what we did was a drugstore, and we knew people were throwing dirty glass into the trash dump. So we thought, well, maybe it'll be clinging to the inside of the bottle glass, but not enough still was. Without the pores of the clay to protect the compounds, it seems to just wash away and be eaten by bacteria and all the rest of it. It seems to really need those pores, whether the pores be from the clay or from a charcoal incrustation. Without those, it just kind of doesn't work at all.
Okay. The next question is from Shikha Rai, and she asks, in an archaeological context, when certain pot forms, they get standardized over a period of time. For example, cooking pots or certain ritualistic pots. There could be different as well as multiple sorts of uses depending upon the society in which these pots are made and used. Having said that, what are the limitations of such studies and what should be the ideal sample size?
Well, that's another excellent question. So sampling is actually, I think, the most important and understudied part of residue analysis. What I usually tell people is to think in terms of what I call the functional group. So the functional group is what you as an archaeologist are looking at to interpret. So say you're interested in comparing perhaps a bowl form to perhaps, a pot form or something. Your functional group would be bowls versus pots, not, for example, the color or something like that. So for each functional group, I usually tell people at least 10 sherds, and 15 is maybe better. Now, of course, you can reincorporate these functional groups in your head, right? So say we have 15 bowl sherds and 15 pot sherds. Those are your two functional groups that you thought of when you were sampling, and this is key. But as we look at it and as we interpret the residues, it may be we discover that, the red ones look really different than the brown ones. Well, we can rebuild those functional groups in our minds and then base it on color. And that will we can change our interpretation as we look at the results. But yeah, I think you need at least 10 to 15 sherds in each of the areas that you think as an archaeologist are interpretively important. And this is actually one of those things I can't help you with usually, unless it's an area I myself have dug in, which, in South Asia is nowhere. So it's up to the archaeologist to determine, what they think is going to be meaningful for interpretation, whether it be form, site location, time, level on the site. I would say the very most common mistake that archaeologists make with this analysis is they don't think in terms of functional groups. They just get one of everything kind of thing. So they've got one old pot and one old bowl, and they'll spread it from all over the site, and it's neat. They've got one of everything, But because their functional group size is really 1, it's not meaningful. We can't make, the only interpretation we can make is there were residues or there were not residues. And we can try in hindsight to produce functional groups, but it's not as good as you as an archaeologist knowing ahead of time what you're really interested in terms of pottery contents.
That's a good one. I think this kind of sampling should be now a part of the research design, much before going into the field. Not just for pottery, but all archaeological material, bones or other kinds of artifacts and ecofacts, that we should think of the sampling way before we enter the field and start digging. So the next question is from Sri Lakshmi. She asks, does it help if residue analysis is done to the pottery along with the content? So if it has some kind of in situ filled content like soil, like you showed one of those potsherds which had soot or soil inside it. So do you analyze the content and the pot?
Yeah, that can be very useful. So when it's mostly useful is, again, when you're pretty sure it's been buried with contents in situ, or you think maybe it was, but you're not sure. So in that case, what you can do is look at the residue within the pot and then the residue within the soil. If they look really similar, then you can be pretty sure that the pot was buried in situ with contents, which is super useful, especially with regards to offerings of different types, burials, caches, things of this nature. If the residue from the soil looks not much like the residue from the pot, then it was not probably buried in situ. And if you're pretty sure that it was buried in situ with contents, then it would also, if possible, be a good idea to look at other sorts of analysis, as in the phytoliths, starch. That would be a great moment to start looking at starch, things like that.
Okay. The next question is from Korshinik Veronikamal. Does the process of condensation, so for example, if we keep the pottery inside a Ziploc cover, there will be kind of, some moisture and then there is condensation. Will that affect the residual analysis? How best to pack pottery if we are sampling for a while?
Well, in general, you don't want it to get moldy for sure. So if you've got it, if you maybe collected it in the rain and then you Ziplocked it up and maybe you left it a little too long and it starts to grow mold, that's not going to be good for, your residue, or probably you, really. So if you must pack it in plastic, then make sure it's dry. But again, a paper bag is really better if you haven't got tin foil on hand. If you do have tin foil on hand, try to leave a little air room maybe, because any surviving residues could be attacked by funguses or microbes if it's permitted to get wet or to condense too much.
I see 2 more questions right now. Okay. The next one is by Dr. Shiv Singh. And he asks, when conducting carbon-based residue analysis, what is the typical chronological time frame that we should consider for obtaining an accurate and meaningful result? Are there specific limitations regarding the degradation of organic materials over time?
This really has to do not so much with time as within residue preservation. So yeah, there might be theoretical limits on, how old a residue might be, but as a practical matter, people have found residues from the very oldest pottery. So for example, Dr. Carl Heron has identified, and I think Dr. Oliver Craig have identified residues from Jimon pottery in Japan. And as long as the residue appears intact, then you can also get decent compound specific data from it. Now, since the preservation of the residue varies so much according to environment, then you would probably do better with pottery from perhaps drier environments and those with more consistent. So Egyptian pottery has great residues in it because, it stays dry and preservation in the desert is so good. Residues from the high Arctic are, you probably won't be surprised to know, very excellent. Scandinavia has beautiful residues because it's fairly chilly and we're not seeing many phase changes. However, residues from where I am in coastal North Carolina are generally not preserved well. The soil is very sandy and very acidic because of the pine barrens, and then saline because of the ocean. So the combination of the salt, the acid, and the sand all together, and then the variations in temperatures tend to mean that residue preservations are very poor in this part of the world. So it's more a regional thing, I would say, than a time-based thing. And I've noticed as well that amounts of residue preservation in pottery vary quite dramatically from place to place. So the traditional measure developed by Evershed for England was, he always used to say, about half the potsherds have residues in it. But as I work in, say, the Midwestern parts of North America, I'm seeing maybe more like 80 to 85%. People in Scandinavia are seeing like 90 to 95%. And I don't think it's that people are using pottery differently. I'm thinking it's that residue preservation is better in Scandinavia than in England, at least in part.
Okay, and the last question, no, I see two more. So Shikha again asks that we consider burial pots as a standard sample to do residue analysis, but do you feel we also need to understand burial conditions of these burial pots, I guess, particularly places like South Asia, where we have so much diversity in environmental conditions, coastal to desert, floodplains to hilly regions. What do you have to say about that?
Yeah, it really is key to understand the conditions in which the pot was buried. So again, I've done a few over the course of my career, several experimental studies, more than many people. And what I mostly found is that there's very great variation. So for example, if I bury pieces of the same pot with the same residue in a very swampy environment, which I have in my backyard, and in a very sandy environment, which I also have in my backyard, and if we perhaps mail them to a friend up in the Piedmont, which we've also done, and have them bury it in their backyard, the residues look very differently in these three environments, and all three will look different from one we put in the freezer to see what it originally looked like. So things such as variations in soil chemistry, acidity, moisture, particle size, all these things have an effect, which is why we have to be very careful when comparing residues from sites with very different environments. Because, the main difference appears to be in just the amount of breakdown and the amount of microbial degradation, and of course, hydrolysis and oxidation. It can shift the interpretations a little, but not so much. Usually it's mostly the amount that's left that varies quite a bit. So yeah, if you were looking at a burial pot from, an area with a very acidic, saline, sandy, soil, and, it was maybe monsoon to dry and the temperature varied quite a bit, I would expect your preservation to be worse than somewhere with a clayier soil and perhaps more moderation in the temperature.
So just two more. I hadn't seen the Q&A box, and there is one question there. So this is an anonymous participant who asks, archaeobotanical and faunal analysis can provide data to interpret whether people consumed a plant-based or meat-based diet. Additionally, the presence of cattle, sheep, goats in faunal records may indicate a milk-based diet. Now, in terms of pottery function, this can be assessed by examining the shape of the pottery. Like some shapes hold liquid, some others have larger surface area and may function as cooking vessels. So when it comes to lipid residue analysis, which is very costly and requires specialized expertise, the addition to the knowledge pool is relatively limited. What is your opinion on this?
Well, you have to be cautious about when you use it. I wouldn't necessarily do residue analysis on every pot you have. But it's worth noting that what residue analysis does do is it sort of verifies the form function analysis, right? So I have done sites in which people thought they had cooking pots, but when we analyzed the residues, they hadn't been used for cooking. And similarly, it can be quite useful actually in terms of whether it's been used at all. Has the pot been used for water, perhaps, or for dry ingredients? And depending on your area, form function analysis, although a useful indicator, is perhaps not always as great as we hoped as archaeologists. So, for example, from a site in, I think it was Louisiana, we did a residue analysis on a bottle, and we sort of assumed, since it was a bottle, that it would include liquid, and we were all set to look for a variety of the drinks that we would expect to find, whether it be in that part of the world a nut-based drink or a caffeine-based drink. That's what we were looking for. And yet, when we did the residue analysis, it wasn't a drink. It looked more like a stewish kind of a thing, with a mixture in this area of meat and plants. And it looked like it had been buried in situ, possibly as an offering. So in this case, which is an unusual one, I admit, the bottle, even though the form suggested a drink, had actually been used for a stew, probably because it was a cash offering. And so in that case, the ability to serve versus not serve was not really key because they weren't planning to serve it. So I would say that residues are useful in terms of verifying our form function analyses. Usually they're right, but sometimes they're not. And also it can be useful in terms of just kind of checking to see what's been used and what hasn't been, which is not always, what we would expect. And I would also say it's not, as analyses go, that expensive. My lab does the analysis for about $81 US a sample, which compared to various other analytical types is not that expensive. It does take a while, and is time consuming and does require technical special equipment, but I would say it's equally as worth doing as most of the other special analyses, as in, faunal analysis, PBOT, things of that nature.
Just one last question. This is by Ramya Saravanan. So she was actually adding to the question that was asked previously on the burial pottery or the burial conditions. So she asks, while excavating the burials, the environment conditions will affect the residues when the trenches are kept open in the dig, right? And the current climatic conditions will affect the residues of these potteries that are exposed to open air. So how can we prevent the damage? So let's say there is a kind of a delay in sampling the pottery from the site for a few days, perhaps, or a few hours. So how can we cover the pottery with something, or what can we do?
If it's only a few days, you're fine. Now, if you're leaving it out a whole season, I would probably take the sample earlier. But I mean, the residues, once they're in the pot, are fairly robust in many ways. I mean, consider some of the samples we've worked from over the years. People dug them up, they had them in plastic, they were sitting in their museum for a while, and we still found residues, often with a lot of plastic in there as well, but they were still interpretable. So I would say if it's only for a few days, just, sample it if you can into paper or tin foil, and it shouldn't make a massive difference. But if it's a very, very long excavation and it's open for, maybe the whole season, maybe take the sample on the earlier side. Yeah.
Okay. I also see a lot of people thanking you for this talk. And I think there's one question on what are C4 plants, but that has already been answered in the presentation. And Debopam Ghosh, the one who asked, I think I would just like to tell him that we will be putting up this video on YouTube, IIT Gandhinagar's YouTube channel, in a few days. So you could listen to this presentation again and clarify your basics on the kind of plants, right? C3, C4. So otherwise, we have covered most of the questions, and thank you so much for very clear, crisp explanations. And it's such a pleasure to listen to you. And I'm sure that especially for the students who attended this talk today, it was quite. I think it kind of clarified their fundamentals on ORA and stable isotopes and categorization of plants and all of that. It's always such a pleasure to interact with you, Professor Reber. Thank you so much for coming. And it's quite early in the morning for you, so I think and also a weekend. So we should probably end the talk now and kind of let you be free. So I also thank all the participants who have joined us and for all the wonderful questions. And on behalf of my colleagues at the center and all the students and Ahana, we thank you and hope to continue with the collaboration. And it's very exciting that you now have, I think, a good kind of good comparable data sets from South Asia as well to add to your own work. So yeah, we wish to really continue with this collaboration in the future and see many more exciting researchers coming out of it.
Thank you. And thank you for your kind reception. Really appreciate it.
Thank you so much. And I'm sure if the students have any questions, maybe they could write to us and we could forward some of those questions to you later on. I can consolidate all the other queries because I still see one or two questions coming up here, but I think we are running out of time and we can always address them later through emails. So,
okay.
Yeah.
Sounds great.
Have a great weekend, everyone. And please join us again next month on the last Saturday for another talk on archaeological sciences, maybe from a different time period, different site, different expertise. So have a good weekend and bye-bye.